Hello students, this is Lohita. Here we are into zoology NEET class. So, in our previous class actually we have learnt about some of the Mendelian disorders, right. So, we have learnt about color blindness in our previous class, right. Hemophilia we have learnt, sickle cell anemia we have learnt. So, today we are going to learn few more examples of Mendelian disorders, okay. So, the first example of Mendelian disorder that we are learning today is phenylketonuria, okay, phenylketonuria, okay. So, phenylketonuria is actually an inborn error of metabolism. It is actually an inborn error of metabolism. Phenylketonuria is an inborn error of metabolism, and sometimes phenylketonuria is also inherited as an autosomal recessive disorder. It is also inherited as an autosomal recessive disorder. Sometimes it is also inherited as an autosomal recessive disorder. That means autosomal recessive disorder means both males and females will have an equal chance of inheriting of this particular allele, okay. So, now students, phenylketonuria is actually caused due to a gene which is defective for the production of enzyme, called phenylalanine hydroxylase, okay. Generally, you know, the importance of this phenylalanine hydroxylase is this phenylalanine, which is an amino acid, is converted into tyrosine using phenylalanine hydroxylase, using phenylalanine hydroxylase enzyme, phenylalanine is converted into tyrosine. But in the absence of this enzyme phenylalanine hydroxylase, because of defect in the gene producing this enzyme, in the absence of this enzyme phenylalanine hydroxylase, phenylalanine is converted into phenylpyruvate. Phenylalanine is converted into phenylpyruvate, okay. This phenylpyruvate is stored in the brain. It is stored in the brain, resulting in mental retardation, okay. And because it is not because it is not metabolized even by the kidneys, phenylpyruvate is excreted through urine. Phenylpyruvate is excreted through urine because it is even not filtered by the kidneys. Phenylpyruvate is excreted through urine. This is all about phenylketonuria. So, this condition of storing of phenylpyruvate in the brain resulting in mental retardation, excretion of phenylpyruvate through urine is condition called as phenylketonuria. Phenylketonuria is actually an inborn error of metabolism. It is an autosomal recessive disorder. Also, a gene defective for the enzyme phenylalanine hydroxylase is generally responsible for this disorder. When generally phenylalanine hydroxylase is converted into tyrosine in the presence of this enzyme phenylalanine hydroxylase. But due to defect in the gene for this enzyme, phenylalanine hydroxylase is not produced. So, instead of getting converted into tyrosine, phenylalanine is converted into phenylpyruvate. This phenylpyruvate is stored in the brain, resulting in mental retardation, and it is not filtered by the kidneys, so it is excreted through urine, okay. So, that is all about phenylketonuria. Next, we are going to learn about thalassemia. Thalassemia is also an autosomal recessive disorder. It is an autosomal recessive disorder. It is because it is autosomal recessive disorder, autosomal, that means both males and female children can be affected with this disorder. Because it is autosomal, both male and female children can be affected with this disorder. Because it is recessive, okay, the children can be affected with thalassemia. The children can be affected, they can be affected only when Only when mother and father both are carriers for the defective gene. Both are the carriers for defective gene. Okay. So thalassemia is an autosomal recessive disorder. Because it is autosomal, both males and females have equal chance of inheriting this disorder. Because it is recessive, children are affected only when their mother and father both are carriers. Okay. So this thalassemia is actually, you know, because of the defective hemoglobin. Because of the defective hemoglobin, the red blood cells are killed. Okay. There is a large amount of degradation of red blood cells with defective hemoglobin. There is degradation or destruction of red blood cells with defective hemoglobin. Okay. So this thalassemia is actually caused due to defective hemoglobin, abnormal or defective hemoglobin. Abnormal defective hemoglobin is responsible for this disorder, thalassemia. Okay. So here during thalassemia, the genes which are producing the globin chains of hemoglobin are affected. Okay. The genes producing the globin chains of the hemoglobin are affected. The globin of the hemoglobin are affected. The genes which are producing the globin part of the hemoglobin are affected. Okay. Depending upon, you know, generally in hemoglobin there will be 4 different type of globin chains. 2 chains of alpha globulin, 2 chains of alpha globin, 2 chains of beta globin. Okay. In hemoglobin generally 2 alpha chains and 2 beta globin chains will be present. You know, there may be defect in any of the globin chains. Okay. If there may be defect in alpha globin chains or there may be defect in beta globin chains. The defect in alpha globin chains is because if the defect is in alpha globin chains, then this type of thalassemia is called alpha thalassemia. Okay. If there is a defect in alpha globin, then this type of thalassemia is called alpha thalassemia. If there is a defect in beta globin, then that type of thalassemia is called beta thalassemia. Okay. If there is a defect in alpha globin, then it is called alpha thalassemia. If there is a defect in beta globin, then it is called as beta thalassemia. So depending upon the type of globin chain affected, we are naming the type of thalassemia. Okay. So let us learn now in detail about these 2 different types of thalassemias, alpha and beta thalassemia. You know, students, there are 2 genes, 2 closely related genes called as HbA1 and HbA2. These are the 2 genes which are responsible for the production of alpha globin chains. Okay. Alpha globin chains are produced due to 2 very closely located genes called HbA1 and HbA2. Okay. Any change in the genes, any change in these genes will result in lower production, any change in these genes will result in the lower production of alpha globin, Okay. If see here 2 genes HbA1, HbA2, right. So there will be 4 alleles which are for this alpha globin, right. 4 alleles will be there for this alpha globin. So more alleles affected, then less will be the alpha globin synthesized. Okay. More genes affected, less alpha globin will be produced. Less alpha globin is produced, that means abnormal hemoglobin is produced. Okay. So coming to the other case, beta thalassemia. Okay. See, beta thalassemia, as I have told you, that it is caused due to defect in the beta globin chains. Okay. So there is only one gene called as HBB, which is located on the chromosome number 11. Okay. This alpha globin genes are present on chromosome number 16. Okay, whereas beta globin genes are present in the chromosome number 11, only one gene HBB which is present on the chromosome number 11. So, this is having two alleles, right. So, according to the number of alleles affected, the number of beta globin chains will be produced. More alleles affected, then less will be the beta globin chain production, less will be the beta globin chains. Okay. So, here because of the less number of beta globin chains than normal, this is going to result in abnormal hemoglobin, Okay. So, here because of the presence of abnormal hemoglobin in both the cases, the red blood cells are killed. The affected red blood cells with this kind of abnormal hemoglobin, they are killed. Okay. So, with the increase in the killing of the red blood cells, it will result in anemia, which is called as thalassemia, Okay. So, once we will revise what we have learnt about thalassemia, students. See here, thalassemia is actually an autosomal recessive disorder. Here generally, because this is autosomal disorder, both males and females are affected, but because this is autosomal recessive disorder, the children will be affected only when their mother and their father both are carriers. Okay. So, here this disorder is caused due to degradation of the red blood cells with defective hemoglobin. So, there is improper hemoglobin produced in the case of thalassemia. Okay. So, there is abnormal or defective hemoglobin produced. So, here the genes which are producing the globin part of the hemoglobin are affected. There are 2 alpha globin chains and 2 beta globin chains in the hemoglobin generally. So, if the alpha globin chains are affected, then it is called alpha thalassemia. Beta globin chains are affected, then it is called beta thalassemia. So, in case of alpha thalassemia, here the alpha globin chains are affected. The chromosome number 6 contains 2 genes for alpha globin. They are HbA1 and HbA2. These are the 2 genes for alpha globin. Any change in these genes will cause changes in the production of alpha globin. More the number of alleles affected for this alpha globin chains, less will be the alpha globin production, resulting in abnormal hemoglobin. Okay. Coming to beta globin chains. Beta globin chains defect is resulting in beta thalassemia. There is only one gene responsible for beta thalassemia, that is HBB, which is located on the chromosome number 11. Okay. There are 2 alleles only for this gene, and more the number of alleles affected, less number of beta globin chains will be produced. Okay. So, here finally resulting in abnormal hemoglobin. So, the red blood cells with abnormal hemoglobin are generally killed, as a result of which the number of red blood cells will be decreasing, that is called as anemia, and this type of anemia because of the defective hemoglobin is called as thalassemia. Okay. So, now students, we need to learn one more thing here. Okay. So, see what is the difference between sickle cell anemia and thalassemia? Sickle cell anemia and thalassemia. See, in case of sickle cell anemia, the quality of the here in case of sickle cell anemia, what is happening? The red blood cells are produced, but they are sickle shaped, right? They are sickle shaped. So, here in this case, there is no change in the quality of the hemoglobin. There is no change in the quality of hemoglobin is same. Right, there is no change in the quality of there is a change in the no change in case of the quality of hemoglobin. Quality of hemoglobin is same, right? Quality of hemoglobin is affected because hemoglobin is becoming sickle cell, right? So, here in case of sickle cell anemia, quality of the hemoglobin is affected. Okay, whereas in case of thalassemia, quantity is affected. quantity of hemoglobin is affected. Okay, that is the difference between sickle cell anemia and thalassemia. Both are the types of anemia only. So what is affected here is important. In sickle cell anemia, quality of hemoglobin is affected, whereas in thalassemia, quantity of hemoglobin is affected. Okay, so that's all about the few types of Mendelian disorders, students. Mendelian disorders, we have studied that they are caused due to one single gene mutations, monogenic inheritance also we are calling it. Okay, so here only one gene defect is there which is resulting in these disorders, and these disorders they are following Mendelian pattern of inheritance. So that is why these disorders are called as Mendelian disorders. We have studied the examples of Mendelian disorders like color blindness, sickle cell anemia, hemophilia, phenylketonuria and thalassemia. Okay, now we have to learn about chromosomal disorders. Okay. Chromosomal disorders. Chromosomal disorders, they are caused due to errors in the number or the structure of the chromosomes, errors in the number or the structure of the chromosomes. Okay, so chromosomal disorders are caused due to errors in the number or structure of the chromosomes. Okay, so you know students, these errors are caused due to different reasons. See, for example, some errors are caused due to failure of segregation, of the chromatids, during meiosis, That means the paired chromatids during meiosis, they won't separate, as a result of which one set of gametes will have extra chromosomes, whereas the other set of gametes will have less number of chromosomes. So here just the number of chromosomes is changing. They may have an extra chromosomes or they may have less number of chromosomes. Okay, so due to failure of segregation of the chromatids during meiosis, some of the gametes they will have extra chromosomes, some of the gametes they will have less number of chromosomes. This condition of change in the number of chromosomes is called as aneuploidy. Aneuploidy is nothing but change in the number of chromosomes. This condition is called as aneuploidy. Okay. So students, due to failure of cytokinesis, cytokinesis means cell division. Cytokinesis means cell division. Due to failure of cytokinesis after meiosis, there will be changes in the number of sets of chromosomes. There will be number of sets of chromosomes will be changing. So as a result of which the chromosomes may be multiplying in sets of chromosomes are changing, right. So this condition is called as polyploidy, and this is generally found in the plants, and in man generally aneuploidy is seen. Okay, so let us learn some of the disorders caused due to this aneuploidy in case of human beings. Okay. So first we will learn about allosomal disorders. Okay. So students, allosomal disorders are nothing but here these chromosomal disorders are caused due to the sex chromosomes. Okay. So allosomal disorders we are learning. The first allosomal disorder we are learning is Klinefelter's syndrome. Okay, Klinefelter syndrome. In Klinefelter syndrome, males are affected. Okay, males are affected with this Klinefelter syndrome. These individuals, they have an extra X chromosome. They have an extra X chromosome. Okay, so the karyotype of these individuals is like a 47 XXY. Okay, it is important to know how we write the karyotype, students. Karyotype is 47 and XXY. So 47 is indicating that there is an extra chromosome. Generally 46 chromosomes are only found in man, right? We all know this. Generally only 46 chromosomes are present in man. So here number is showing 47. 47 because there is an extra X chromosome. Okay, so these are the males which have an extra X chromosome. Here in this kind of individuals, male characters are shown. Male characters are shown, but the feminine characters are not completely suppressed. Okay, they are not completely suppressed. So these individuals, they will show development of breasts. These individuals, they will be showing development of breasts. Okay, these females, they will be showing development of breasts. This condition is called as gynecomastia, and they also show, in some cells they also show bare bodies. In some cells. Okay, so these are the individuals, like these are the males with the bare body. So these are the males with the bare body. Okay, so these affected individuals exhibit hypogonadism. That means their gonads are not developed properly. And generally the affected individuals are sterile. This is all about Klinefelter syndrome. Klinefelter syndrome, males are affected. So they have an extra X chromosome. The karyotype will be like 47 XXY. So the male should have an XY only, but here XXY, one extra X chromosome is present. So here the male characters are shown, but the feminine characters, the female characters, are not completely suppressed. They show development of breast called as gynecomastia, and they also show bare bodies in some cells. So Klinefelter males are the males with the bare body. Then they also exhibit hypogonadism and they are generally sterile. Okay, that's all about Klinefelter syndrome. Now we are going to learn about Turner's syndrome. These are the individuals whose karyotype will be like, whose karyotype is 45X. The karyotype of these individuals is 45X. So this Turner syndrome is affecting the females. These females are without a bare body. These are the females without a bare body. Generally females, they have a bare body, but these are the females without a bare body. They don't have a bare body because they have only one X chromosome. Generally females have two X chromosomes, right? But these are the females with only one X chromosome. These individuals are the females with only one X chromosome. So they are without bare bodies. Okay. So this Turner syndrome affected female shows stunned growth, webbed neck, broad shield-like chest, and this individual is going to show gonadal dysgenesis. gonadal dysgenesis. That means there will be no proper development of the gonads. Okay, that's all about Turner syndrome. See students, Turner syndrome, here the karyotype is 45X. These are the females which are affected. These are the females without a bare body because they are having only one X chromosome. There is only one X chromosome in these affected individuals. Here we will be observing that there is very stunned growth in this affected person, and they will be showing a webbed neck. Webbed neck, that means their neck will be having like some tires on the neck will be seen. They have a broad shield like chest, and gonadal dysgenesis is also shown. Okay, so that's all about allosomal disorder, Turner syndrome. So here we have learned about the two allosomal disorders students. We have learned about Klinefelter syndrome and Turner syndrome. Here in both the cases the sex chromosomes are affected, allosomes are affected. Klinefelter male is the male with an extra X chromosome, and Turner's female is the female without one X chromosome. She is having only the female with only one X chromosome. So Klinefelter male is a male with a bare body. Turner's female is a female without a bare body. Generally, bare bodies are found in females. Bare bodies are those structures found in females which are helpful in identifying the sex. Bare bodies are generally indicating the presence of the number of X chromosomes. Bare bodies, they generally indicate the presence of number of X chromosomes. Okay, bare bodies, they generally indicate the number of X chromosomes. Okay, they generally indicate the number of X chromosomes. That's all about allosomal disorders. Now we need to learn about the autosomal disorders. Autosomal disorders are generally caused due to autosomal disorders are generally caused due to changes in the autosomes. Okay, they are generally caused due to changes in the autosomes. Okay, so students, they might be having an extra chromosome, or they might be having a chromosome less. So trisomy is a condition where there is an extra chromosome, 2n plus 1. This condition is called trisomy. There is an extra chromosome. Monosomy means there is one chromosome less, 2n minus 1. This is monosomy, one chromosome less. Okay, generally trisomy and monosomy are found in case of autosomal disorders. Okay, so these autosomal disorders are caused due to changes in autosomes. They exhibit trisomy or monosomy. Okay, so these autosomal disorders are generally common in those children who are having the mothers having children at very old age. So the women having children in the very old ages will have more chances to give birth to those children with autosomal disorders. Autosomal disorders are very common in those common to the children of those mothers who are having children at very late stages. Okay, let us see some examples of autosomal disorders. First, we are going to learn about Down's syndrome. Okay, Down syndrome is caused due to trisomy of 21st chromosome, trisomy of the 21st chromosome. Okay, so here we can simply write for autosomal disorders, we can simply write trisomy 21. This is very clearly indicating that there is a presence of an extra 21st chromosome. Generally, any chromosome will be represented in pairs, right? In human beings, generally any chromosome will be represented in pairs. Every chromosome will be occurring in number, but here trisomy means one extra chromosome will be present. So generally, other than two 21st chromosomes, there will be an extra 21st chromosome. There will be an extra 21st chromosome. Okay, Down syndrome was first identified by Langdon Down in 1866. Langdon Down has identified Down syndrome in 1866, and after his name only this Down syndrome has got its name. Here during Down syndrome, physical, psychological and mental retardation occurs, and mental retardation will occur. Okay, in this condition physical, psychological and mental retardation is very common. The affected individuals with Down syndrome will be very short in height, they will have a very short height, they are having a small round head, they have a small round head, they have a partially open mouth, they have a partially open mouth, and they have these affected individuals, they have clenched palm, okay, these are some of the effects, and fissured tongue, fissured tongue. These are some of the features which are shown by the affected individuals with Down syndrome. Short in height, round head, partially open mouth, clenched palm, fissured tongue. These are some of the characters shown by Down syndrome. Down syndrome is due to trisomy 21. Here we are observing that there is an extra 21st chromosome. It was first discovered by Langdon Down in 1866. It is causing physical, psychological and mental retardation. Short, round head with partially open mouth, clenched palm and fissure are some of the characters shown by these affected individuals. Okay, these are some of the characters shown by these affected individuals. Okay, so let us learn now how the karyotype of this individual is written. Karyotype. Okay, so karyotype of this affected individual is written as 47 plus 21, 47 plus 21. So, here 47 is indicating that the number of chromosomes from 46 has changed to 47, plus 21 is indicating that because of the presence of extra 21st chromosome. Okay. So, in this way we will be writing the karyotype here, it is important. Okay. Next we will be learning about Edwards syndrome. Edwards syndrome is caused due to trisomy of 18th chromosome. Trisomy of 18th chromosome. That means due to the presence of 3 18 chromosomes. Generally 2 18 chromosomes should be present, right? But here there will be 3 18 chromosomes. Trisomy of 18th chromosome results in Edwards syndrome. Okay. So, there are many different complications in Edwards syndrome, particularly here in Edwards syndrome, cardiovascular abnormalities, cardiovascular system abnormalities and kidney abnormalities are occurring. Cardiovascular system and kidney abnormalities. Cardiovascular and kidney abnormalities are generally affecting in these people. Okay, because of the very severe complications in cardiovascular system and kidneys, these children, they won't be able to live long. They cannot live for a long time. They cannot live for a long time. Okay, the karyotype of this individual will be, the karyotype of this individual will be 47 plus 18. Okay. Then we have Patau syndrome. Patau syndrome. Patau syndrome is caused due to trisomy of 13th chromosome. Okay. So, the karyotype here we can write it as 47 plus 13. Okay. So, Patau syndrome, there are many complications, students, in case of Patau syndrome. They showed abnormalities in the cardiovascular system, kidney related abnormalities, abnormalities related to the brain and spinal cord. Okay, brain and spinal cord. They have very small eyes. This condition is called as microphthalmia. Okay. So, due to all these complications, the Patau syndrome child also will die within few days to weeks only, he will be dying. Okay. So, that is about Patau syndrome. Trisomy of 13th chromosome, karyotype will be 47 plus 13, cardiovascular system, kidney, brain and spinal cord are the parts which are affected in this case. These are the children whose eyes are very small. This condition is called microphthalmia. They will just die within few days or within few weeks after birth itself because of so many complications in their body. Okay. That is about Patau syndrome. Now, we have another syndrome called Cry-Du-Chat syndrome. Okay. Cry-Du-Chat syndrome. which is also called 5p minus syndrome. 5p minus syndrome. See students, this cry-do-chat syndrome, 5p minus syndrome, is actually caused due to deletion of the short arm of 5th chromosome. Okay, this cry-do-chat syndrome is caused due to deletion of the short arm of the 5th chromosome, deletion of the short arm of the 5th chromosome. Okay, cry-do-chat syndrome is caused due to deletion of the short arm of the 5th chromosome. Here, you know, we cannot call this as monosomy. We cannot call this as monosomy. We cannot say it as 5 monosomy, because there is no 5th chromosome is not completely deleted. Only a part of the 5th chromosome is deleted, right? Only short arm of the 5th chromosome is deleted. Because of that, we are not calling it as monosomy. We are not calling it as monosomy. Instead, we are calling it as 5p minus syndrome. Okay, and the karyotype of the affected individual will be written as 46 minus 5p. 46 minus 5p. In this way, we will be writing the karyotype of the child with cry-do-chat syndrome. This name cry-do-chat syndrome is actually given because this word cry-do-chat is actually having a French meaning. This word cry-do-chat is actually having a French meaning, that is cat like cry, cat like cry. Okay, so this child with cry-do-chat syndrome will be crying like a cat because of his problem with the larynx and nervous system, because his larynx and nervous system are affected. Because his larynx and nervous system are affected, he will be having a cat like cry. Okay, in this child there are some other complications also, like the presence of cleft palate and the persons will have a short head with abnormal facial features, short head with abnormal facial features. They will be having a short head with abnormal facial features. Okay, that's all about cry-do-chat syndrome. It is 5p minus syndrome caused due to deletion of the short arm of the 5th chromosome. The karyotype will be 46 minus 5p. French, it's a French word. Cry-do-chat is a French word which is having a cat, which is meaning a cat like cry. The affected child will be crying like a cat because of his larynx and nervous system problems. He is going to have a short head with abnormal facial features. With all these conditions, this child will also not live for a very long time. He will be dying within few days after birth. Okay, so that's all about cry-do-chat syndrome. So, the last disorder, last autosomal disorder in this genetic disorders we are learning is chronic myelogenous leukemia. Chronic myelogenous leukemia. This is caused due to translocation. This is caused due to translocation. Translocation means changes in the parts of the chromosome. Okay, this is caused due to a reciprocal translocation. This is caused due to a reciprocal translocation between the chromosome number 9 and 22. Okay, so a part of chromosome number 9 is exchanged with a part of chromosome number 22. Some part of chromosome number 9 is given to 22, 22's chromosome part is given to 9. Okay, this results in abnormally short 22nd chromosome and abnormally long 9th chromosome. Okay, so this abnormally short 22nd chromosome is called as Philadelphia chromosome. Okay, this abnormally short 22nd chromosome is called Philadelphia chromosome. Because this Philadelphia chromosome is formed due to translocation between 9 and 22, this translocation is called as Philadelphia translocation. This translocation is called Philadelphia translocation. Okay, so because of this Philadelphia translocation, from chromosome number 22 from chromosome number 22 an abnormal enzyme called tyrosine. An abnormal enzyme called tyrosine kinase is formed, okay. Because of this translocation from chromosome 22, an abnormal enzyme called tyrosine kinase is produced. This tyrosine kinase is causing unregulated cell division, okay. It is causing unregulated cell divisions. It is causing unregulated cell divisions leading to cancers called as chronic myelogenous leukemia, okay. So, that is all about chronic myelogenous leukemia. Reciprocal translocation between chromosome 9 and 22 will result in an abnormally short 22nd chromosome and an abnormally long 9th chromosome. So, this abnormally short 22nd chromosome is called Philadelphia chromosome. This translocation between these two chromosomes is called Philadelphia translocation. This abnormal 22nd chromosome will result in the production of enzyme called tyrosine kinase, which causes uncontrolled cell divisions resulting in cancers called as chronic myelogenous leukemia, okay. So, students, that is all about this concept genetic disorders. With this we finish to learn even the principles of inheritance and variation, okay. Thank you all for joining the class today. We will meet again in the next class. We will start a new chapter in the next class. Thank you all.